Well, happy Friday, everybody. We're almost to summer. And after a few really good talks from Europe, we come back to the Great Lakes in today's talk. And maybe before I introduce Brian Weidel, today's speaker, I'll just note that we do not yet have a talk scheduled for next month. So if anybody out there is interested in participating in this, what we think is a really good venue to share your results and to generate discussion, please reach out to Nick Boucher or to myself in the next couple of days. If we don't get any volunteers, we may try to wrangle some volunteers, so to speak. So we're looking at July 8th. So that'd be 12:30 p.m. Eastern time, July 8th. So be thinking about that. So now to introduce today's speaker, Brian Weidel. Brian actually grew up in Pennsylvania, I believe, got his bachelor's at Cornell University, where he also played football, which is a pretty cool thing, I think. He stayed at Cornell, got his master's degree, and then got his PhD and did a year of postdoc at the University of Wisconsin in Madison. So his advisors included Cliff Craft, Chuck Kruger, and Jim Kitchell. We were fortunate enough to recruit and hire Brian Weidel to USGS Great Lakes Science Center in 2010, where he has been at the Lake Ontario Biological Station. But as you'll see from today's talk, Brian goes outside of Lake Ontario to do some of his research, and certainly his research has implications for beyond that one particular lake. So today he's going to talk about lack of cracks. How does the habitat influence Lake Ontario coregonine spawning success? He says the talk is about 30 or 40 minutes. And as we normally do, hopefully you'll stay on and participate in a set of engaging discussion following his talk. So thanks, Brian.
Absolutely. Thank you, Bo and Nick. Okay, everyone hearing me okay? Great. All right. So, yeah, I want to first acknowledge and thank you all for taking your time on this. Well, it's beautiful here Friday afternoon. And quickly acknowledge just an absolutely great team of individuals and folks who've contributed, co-authored, PIs, et cetera, on this work from across the basin. There's more have contributed, but it's just been really fun to tackle this question that we think is actually really important in terms of coregonine restoration in Lake Ontario and maybe other systems. As Bo mentioned, we'll talk about Lake Ontario. I'll get into some of them, why we're looking at this question, how we're looking at this question, some of the methods that we've developed to do it, and our results from Lake Ontario, and a little bit of our results from Lake Superior as well. Let's see if we get everything working here. So first, to acknowledge and thank Great Lakes Restoration Initiative. We've been fortunate to get funding through a variety of different means in that group, and then just in-kind support from a whole bunch of different tribes, agencies, and academic organizations. I'm going to lead off to try to keep myself, there's a lot of different topics that I want to talk about with this, but really a take-home message is first is that we are hypothesizing that anthropogenic changes to spawning and incubation habitat are an underappreciated driver of coregonine populations in some lakes or some regions. We think that even remediating that habitat might be an efficient restorative action, but for that to be true, we really need to understand what aspects of that habitat, what mechanisms, what components, what needs to be remediated. In other words, thinking about sort of like what kills more eggs? Is it the physical disturbance? Is it eggs being moved, temperature changes, sedimentation, light, climate, oxygen? Certainly it's a host of those, but we need a ranking, right? And so we're proposing, and we're using, and we propose to use a life stage assessment for restoration. That's sort of our approach, because we see it real critical to not just list what a potential impediment is, but to rank it, right? For a management agency or group to make use of finite resources, you really need to understand how much bang are we getting for our buck for different manipulations or actions we can take. So really ranking that is key, and that's what we're trying to go after. In so doing, we're focusing on the Lake Ontario as our study system. A brief introduction for those that are maybe not familiar. The Lake Ontario watershed as the lower settling basin, as some of my colleagues in the upper lakes refer to it. We are at the bottom of the watershed, smallest lake, very deep, but huge watershed to lake area ratio, so high turnover time and much higher productivity levels than some of the other deep Great Lakes. From a coregonine standpoint, Cisco and lake whitefish remain, but are rare. With bloater, kai and short nose, there's maybe some disagreement about potentially other species out there that were originally in Lake Ontario. Bloater is being reintroduced right now from gametes from Lake Michigan, but kai and short nose have been extirpated. The lake is dominated by non-native species, we can say, and the most abundant of which is the alewife. These scenes that were so familiar to some of even Michigan and Huron lakes still happen in Lake Ontario, although not nearly as frequently. But upwards from a prey fish standpoint, 90% of the biomass is dominated by alewife, with the remaining components made up by rainbow smelt, round goby, and deep water sculpin. The other thing to really understand about this system is, I'll be showing Cisco, but it applies to lake whitefish in general, is they're both declining and rare. Here, looking at the commercial catch of, I think this is Cisco's and chubs kind of lumped together. But from the 1900s on, you see this real steady decline over time, punctuated by maybe a few blip ups. More recently, we tag on the sort of contemporary surveys, either Canadian gillnet surveys or US bottom trawl and the commercial catch, and we see that continued decline. And I think one thing that's important to keep in mind is, right, like, you know, this lake has been changing a lot since the early and mid 1800s. Alewife were introduced in 1860 and, you know, suggested to be most abundant by 1890. A lot of land use change going on. And so the other thing to think about in these time series is, at any point in time, if you're looking at only a 20, 30, or 40-year period, you have these periods of really sporadic but increased reproduction, right? And what's important to keep in mind is, while those are happening in a local time scale, they might seem important. If you zoom back out and take a broader decadal or century scale, you see really they're just kind of blips on a steady decline. And so that really sets the context for why management agencies, the Lake Ontario Committee, which is made up of the state of New York and the province of Ontario, specifically list management objectives to restore either historic spawning populations or increase the remnant populations of Cisco and lake whitefish. One of the other reasons we want to study it in Lake Ontario, what I call the sort of stock and hope approach to species restoration is 0 for 8, depending on what you want to throw into that list. Be it Atlantic salmon, lake trout, lake whitefish stocking had happened really since the early 1900s, Cisco, bloater. We really haven't gotten any of the species that we've tried to restore going with a natural self-sustaining population. The only one that did came back on itself, the deep water sculpin. And so we want to learn from those previous attempts. They can teach us a lot about restoration as we sort of move into this new phase with coregonine, especially lake trout. That's where there was a lot of dedicated effort and really good success in building a diverse population of adults. But generally, natural reproduction hasn't occurred. And so what we want to take away from that is really thinking about restoration at an entire life cycle perspective, and just building up a population doesn't help if there's an impediment blocking somewhere down the way. The other thing is, because of that, our management actions, at least on the US side, are somewhat limited. Stocking's been historically ineffective. There are no sport or commercial fisheries to regulate for coregonines. Without any fisheries, there's no way to regulate or create refugia. And so in terms of levers that an agency or group might pull for manipulating the situation as it is, altering the habitat or conditions is really what it comes down to. And so as we look at that and we think about how previous manipulations and habitat restorations have worked, a lot of things where if you can do small improvements at the egg survival stage, you can often have large changes in the population. Not always, but often. Finally, another reason is just the unbelievably small empirical understanding we have of coregonine spawning. I often ask folks to pick any other freshwater species of fish or group of fishes, and that's so important to society that we know less about the spawning, right? Bass, Pacific salmonids, pike, bluegill. Compare that to coregonus, and we just don't have the empirical evidence. And a lot of that comes from simply when they spawn, right? A lot of this is like a November, a December, a January, even later, and then an overwinter incubation period. Our gears, our facilities, we're not set up to do that. We think that has created sort of a real sort of lack of knowledge or knowledge gap that prevents us from understanding the entire life cycle of these organisms. And then finally, there are definitely different ideas about what constitutes spawning habitat, and there's for good reasons. There's evidence that from Lake Superior, Dreyer and Beal in the 60s stated that then Lake Herring and Cisco show no preference for a particular bottom type. Whereas 30 years prior, Pritchard in Ontario sort of definitively stated Cisco eggs are deposited in crevices of the rocks and waters from 2.5 to 3 meters deep. What was really neat is some of the early elements of the project that I sort of now working on. Matt Pavey in his 2022 paper, he actually studied and tried to apply the same metrics to different Cisco populations across the lake. And he found evidence that supports this idea that the behavior or the deposition differs across lakes. And so that's important to keep in mind, but we want to understand it at both the local and the regional scale. And that's because, you know, as we've taken on this Great Lakes coregonine restoration, we've developed a framework, and this was adopted by the Council of Lake Committees of all the management agencies in 2018. And it's your adaptive management cycle, but with some nice, clear definitions of what we need to do to get around from the planning to the action phase to learning adjustment and going through that wheel. One of the things that's been so great about this is this gives us a coordinated map, right? You know, folks may differ on pieces of it here and there, but it's hard to really disagree on the overall approach, because where it's been done, it's been successful. And, you know, if you jump into any piece of this, for instance, the contemporary populations, this is something we started in Lake Ontario back in 2018, where we wanted to map the contemporary population where spawning was occurring. We knew where it had historically occurred from sort of anecdotal evidence. Hannah Schaefer did a really nice job in her master's project of putting together information from Goodyear and other sources to see what the history was, but we didn't know what contemporary was. So we used the big collaborative effort of CSMI to go out and collect about 1,000 larval samples in a span of about 3 weeks, about 12 different offices and 8 agencies and academic institutions. And Taylor Brown's master's thesis and subsequent publication showed just how spatially disparate the successful spawning was. Here, the colored points show where and the density that larval Cisco or like whitefish were caught. And the pattern is striking, just how much or how important that northern embayment, that northeastern portion of Lake Ontario is. When Taylor looked at sort of all the potential variables, the one that always came out was that this is the region where ice still forms regularly. Another thing to really take out of that is 94% of those larvae captured were Cisco, right? And some of us have been really kind of trying to draw attention to lake whitefish as a potential conservation concern in Lake Ontario. We don't want to be working on these species and maybe get distracted by the fun idea of bringing in new deep water species while we almost lose one of the key shallow waters. The other thing that was really critical, especially to management, was the fact that reproduction is still, albeit incredibly low levels, still happening in southern shore embayments for both whitefish and Cisco. Now, whether these are remnants, whether they're strays from the Northeast, or even results from some stocking that occurred in here, what it demonstrates is that these populations have not been able to build, and we really think that is attributed to habitat changes and differences among that region relative to the northeastern region. And so that changed management, just jumping in and following the coregonine framework has really already changed at least New York's management perspective on how they go about researching and working on coregonines in Lake Ontario. So I mentioned that we're trying to take a life stage assessment for restoration. And this circle has on the right side what I think a lot of us think of as the portions of the life cycle that we typically study or target: larvae, juveniles, and adults. A lot of the work goes into juveniles and adults out there in assessment. But what we're focused on from a spawning habitat perspective is these other stages and processes: egg deposition, post-spawn egg distribution, and larval emergence. We see them as three distinct stages where if we can observe them in the habitat, then we can make inference on these processes of interest: spawning, egg settlement, and egg incubation success. The problem is, if you think about the methods that we have, they're so much more developed for catching juveniles, adults, and larvae, and the infrastructure that it is for doing all this work on these different stages through the ice, et cetera. And so to do that, we've needed to actually develop quite a bit of methods focused on coregonines. And I haven't been talking about that much in other talks, so I wanted to go in and give folks some understanding. We spent a lot of work on thinking about what we want those methods to be. We definitely want them to be quantitative. We want eggs per meter squared, not presence absence. We want them to be habitat specific, discrete point samples that we can then, get gradients over our habitat. We want to quantify those differences in physical conditions and physical habitat in those different sites. So we need to make sure we have those methods figured out. And then finally, everything needs to be efficient. These are big, spatially diverse processes that we know we need lots, hundreds, if not thousands of observations from. That efficiency, this is a good example. This is a furnace filter mat, which is, I learned about how to do this from Dr. Ed Roseman and Robert DeBruni. And these mats work by entangling eggs. And this is the situation we're faced with in Lake Ontario in the Chaumont Bay, where one of these mats, to get a quantitative measure of that, I think this mat probably took an hour to try to get all those eggs out and deal with it. And so I had a bunch of technicians that were threatening to quit if we didn't come up with a better way. And so we thought about it. We reached out to a lot of folks, the Ellen Marsdens of the world, and just a bunch of Ed Roseman on what can we do? How can we design an egg trap that meets our needs? And we came up with this metal ring design. This is a heavy metal ring. It's boat settable. It's designed for high retention. It's quantitative. That ring sits on the lake bottom. You can see there in the bottom picture, and that mesh sock hangs down or just lays flat underneath it. But then when you retrieve it, all the eggs are captured in there. and easily removed. It's got a couple different layers of mesh to both exclude predators and retain eggs, prevent physical disruption. It kind of works like a sediment sampler would in limnology. And thankfully, I've had some really great colleagues and staff that have been willing to put in a lot of time building. I think we're probably up to have built about 1,000 of these to date. So lots of sewing there. You can see Teresa Bauer is our local seamstress extraordinaire. But we've also tested these gear in the laboratory. We used whitefish caviar and rehydrated it till it had the same density it would as a natural egg. And we put it in tanks along with the fiber mats to test its catchability. If we have a known density of eggs in these tanks, represented by the red bars there, the red lines, how well do the traps sort of recreate those densities? So what we would do is we put the eggs in, not on the traps, next to them, disturb and really stir up the traps. Eggs all move up into the water column and settle back down. When we did that at densities, we found both the fiber mat and the metal ring traps generally did a fairly good job of capturing the densities. Close, obviously, tended to be on the lower side, but this was encouraging to us because it said, okay, we can go out and measure these densities with a fair degree of understanding that what we're seeing is actually what is happening in nature. When we did that for an egg retention experiment, where we put a known number of eggs on top of each egg of the gears, and then we disturbed the substrate, this is where we saw the metal ring design really kind of shining, where the proportion of eggs retained is just a lot higher. And that stands to reason, right? The mats work by the potential of like getting those eggs tangled down inside of them. If that doesn't happen and waves wash them off, that's where you're going to lose eggs. Whereas the metal ring has a bunch of different sort of aspects and attributes that are meant for retaining whatever settles on top of it. Yet when we went out in the field, we saw an interestingly different result. This is work that Cam Davis and Hannah Chance and I did, where we tried to pair observations of the mats and the metal ring traps about 15 meters apart at most, but on the same substrates. And here we found those furnace filter densities were substantially higher than their corresponding metal ring trap. We think this might be, Jason Fisher talks a little bit about sort of the role of using these furnace filters and how they're catching eggs moving along the substrate. And we think that might have something to do with it. We use that folded over design, and a lot of the eggs get caught in that sort of that void between the fold and the mat, but it's definitely something we're paying attention to, in terms of, we don't think we have this all figured out. We want to keep using and having other folks experiment with different gears to try to see what ultimately comes out the best. So then we've also tried to learn from lake trout emergence traps. So pictured here are a couple different trap designs and different ways folks have figured out to deploy them. We like these designs. We wanted to learn from them. We wanted to be able to make our own, make a lot, have high bottle retention. Ellen Marsden often mentioned that being an issue with lake trout. We also needed to design them with a smaller mesh due to the smaller size of coregonines. What we came up was with this sort of Frankenstein somewhat looking design. If that funnel looks kind of familiar, it is actually a 12-inch diameter, like restaurant style strainer with the handle cut off, that we then outfitted with funnels, and we screened the inside and weighted it with bricks. We were fortunate enough to be able to test these traps down at the Tunison Laboratory for Aquatic Sciences in Cortland, New York. The technicians there made space for us really quickly, and it was an awesome working environment where we could allow Cisco that we're about to hatch underneath these traps They were kind of contained in a petri dish. And the guys went in and counted both what was caught in the trap and what was out in the water column every other day for 2 weeks. And we found the traps performed really surprisingly well. On average, they caught 73% of those emerging Cisco larvae underneath them actually ended up in the bottles. We checked it too. We wondered, do we really need that screen on the inside? Well, yeah, we put like let's say it's maybe a 200 micron screen in there, because those emerging Cisco, at least some of them, can be as small as 1 millimeter wide. Well, these things had 3 millimeter holes, and they were just swimming right through the holes instead of going up into the trap. And finally, our efforts to kind of tweak the bottle design to higher retention showed that these things did retain larvae at least for 24 hours at a very high rate. Special thanks to those folks. These guys pulled this work off during the absolute sort of insane portion of COVID lockdown and rules that were coming down on us. And they did so with a smile on their face, and we got these results super quick. So big thank you to that group for all their efforts. Talk about not just the fish element, but characterizing the habitat. This, we're really trying a bunch of different things. We're using drop cam video to record. We can see that drop cam at sort of a live image up on the top of the boat. So we can see as we drop it down what substrate video we're recording. And then we take that back and we categorize that according to the coastal and marine ecological classification standard, which I think has been adopted as a Great Lakes standard by NOAA and other organizations. We've added an interstitial grade to try to get it sort of embeddedness. I'm not going to be talking about that today. We're working with a bunch of different reef assessment groups out there to try to figure out what's the best way of trying to grade interstices or embeddedness. We're also, I wanted to throw in a picture. Pete Esselman and a bunch of his group, Anthony Arnold, they're using multi-beam sonar to get really high resolution bathymetry that Mike Slattery then used a benthic terrain module to classify the benthic terrain. And, we're able to have images like this of our shoals where we can see things at scales that were just not previously possible. You can see the cool cracks in the bedrock and the different aspects and slopes. So we're really trying to apply a bunch of different techniques to understanding the substrate as well. One of the most recent additions we did is we created this physical buoy, as we refer to it, which is meant to help quantify the conditions experienced on a spawning shoal during spawning, incubation, and emergence. And these work with fairly inexpensive pendant data loggers. They're anchored to the bottom, and then there's this float that suspends this pendant, and it can be moved by the wave. So you have a wave come by, it moves that float, and that pendant G logger actually measures those G forces, and this gives you an index of wave exposure. We also add a temperature and light logger to these heavy pavers, so we're getting some indication of other physical conditions on the bottom. If you want to learn more about that, I'll point out Dave Figursky's 2011 L&O methods paper that talks about how you use these G loggers. So we are fortunate, our dive team went out and collected our physical buoys for the first time. So this is kind of, a few week-old data we just got back that we're excited about. We set these things out in a bay, not the primary bay we're working on, but another bay we're interested in doing restoration on. And we wanted to say, all right, we think this is a high disturbance site on one island, and back behind the island would be a low disturbance site. So we set these G loggers or physical buoys at 3 different depths at each one of those sites. And we got some really neat results right off the bat. So here the y-axis is an index of water movement, and those points represent sort of the higher they are, the more movement there is of that float, which we interpret as more physical disturbance, more wave action. What's neat is you can see we kind of got it right. Our higher disturbance was a little bit higher than our lower disturbance. But what's really neat is you can see what we interpret as the onset of ice and the cessation of that disturbance and how it differs by site. And then how also, as you step down in each depth, how the total effect of that water movement declines, right? We know waves have a bigger effect shallow than deep, but now we have a way to actually quantify that in a similar way. If we overlay the water temperature data in red with the light data in blue, we see that those correspond very well. Once that water temperature dipped and that water froze, that corresponds to that period of no movement and also lower light penetration. So we know Taylor Stewart, Mark Vincent, Jason Stockwell, and others are really interested in focusing on how light might influence egg development. Nikki Berry at Ohio State, or Miami of Ohio, is thinking about what light might play, you know, with less ice, less light, or more light, how that might play a role. So we wanted to try to incorporate light in there, and it seems to have worked. All right, so with all these methods developed, what are we actually understanding about the differences in those processes or in those stages of deposition and emergence? The study site we're going to be working on here is Chaumont Bay in the upper right-hand corner of this map. This is a hot spot, to borrow Taylor's map and Taylor Brown's map, and show this is really a very high, a place where high larval densities are consistently found in Lake Ontario for both Cisco and whitefish. And what we see in these figures are deposition rates on the y-axis by depth on the x, and the color of the point represents the substrate. What I guess is a couple things I want to point out first is the order of magnitude, 3 orders of magnitude difference between the Cisco egg deposition and the lake whitefish deposition. We use 2.8 millimeters as a length cutoff to assign the species, and since those are really the only 2 coregonines that we have in Lake Ontario, it becomes easy. Another thing you see is eggs get everywhere at a low density, but there are these hot spots, and also it seems to be the highest densities are in and around those mussel and rock substrates, and that's both the same for Cisco and for lake whitefish. If we look at that data, I'm going to flip it a little bit here just to correspond it to the emergence data. So this is the same data, and this is just for Cisco now. The deposition here, mussels are now illustrated in red, silts in blue, and rock is in black. If we compare the pattern we see in egg deposition, it looks strikingly similar to the pattern we see in the larval emergence data. Where those highest deposition rates we found in those dracaenid shells or dracaenid hash piles is also where we saw the highest larval emergence, and that's in larvae per meter squared per day. The rest of the larval emergence we observed on the rock, cobble, boulder substrates, and then we didn't observe any emergence out deeper, kind of basically out beyond about 5 meters on the siltier substrates out there. So this is where we get this interpretation that interstices seem to matter for Lake Ontario coregonines, or at least Cisco at this point we have evidence of. And that fits with a basic life history model of a fish that's putting its eggs somewhere where they can be protected over time. A couple other things, like what's really happening with those dracaenids? Are these fish targeting those substrate regions, or is it just by chance that wherever the dracaenid shells end up, like there's a local settling area that happens to be where a lot of eggs get washed to? Or conversely, is it the actual structure of the shells? You can imagine a whole pile of empty dracaenid shells, 10 to 20 millimeters long, creating a really sort of rough, full of interstitial space substrate that catches and holds a lot of eggs, you know, an egg that's 2 to 3 millimeters wide. So another thing to point out is, like, what's up with the silt? I say it stinks because it actually does. There's high organic content in those sediments, and eggs on those substrates, at least at the observation level we have here, have yet to produce emerging coregonines. And then finally, again, these larval fishes are genetically barcoded for identification. Despite catching a bunch, we did not catch any lake whitefish. And we set these traps in the areas where we thought we would. So, another nod that we might have a reproductive issue with whitefish in Lake Ontario. A different way to look at the egg deposition data just from above that shows how things are. The eggs appear to be distributed across the embayment there on that left figure, but the highest densities, the terrain colors of green show high density, and then the purple and blue triangles show the highest densities. They all occur on the shallowest regions of either a point or an offshore shoal, but what they really avoid the 0 points out in that sort of deep, which ends up being the siltier areas of the lake. You can see on the substrate map there to the right how those shoals are the primarily rock and or mussel regions in the lake. In 2021, COVID kept us out in 2020, but 2021, we went back out specifically with the goal of testing that our assumption that most of the spawning occurred in the bay and not out in the lake was valid. And we set a bunch of hundreds of traps out in the main lake as well as in Chaumont Bay. And the results really affirm what we saw with larvae, is that the bay is just a larger spawning aggregation or area where they're having success with their spawning than the main lake. Where we did find coregonine eggs in the main lake, they were in that same 2 to 10 meter range over rocky interstitial laden substrates. So one of the things that I stated at the beginning is we want to try to get at the mechanisms that are really driving these changes in depositions or emergence, but we can't separate this correlative element of substrate and depth. Where we see rocky substrates or mussel substrates is over limited depth ranges, right? The boulder, the cobble, the bedrock, and the mussels are all basically 5 meters and shallower, excuse me, then most of the sand and silt are out deeper than that. So really, we can't, it becomes difficult to tease out whether the interstices are important, the depth, or is it really some of both? So our conceptual model as we're moving forward this is that Cisco and whitefish are targeting regions with interstitial substrates. A colleague recently said, are we giving these fish more credit than they deserve? Are they just following some scent cue or some change in slope? That all may be, but really the inference is the deposition appears highest on those regions. Such regions, at least in Lake Ontario, are rare due to sediment, maybe mussel infilling, and they tend to be shallow because we think they're kept open. Those interstices are maintained by the wave action. We think that while the deposition appears highest in those regions, the majority of those eggs are transported downslope to, we'll just say, less than ideal locations. And this has been shown by a variety of folks in other systems. And the way we understand that is when we observe traps with centimeters thick of eggs inside the trap and look at the surrounding substrate, like this image here, there's virtually no eggs. So either these eggs are being moved, predated, or some combination of both. We know there's high egg predation in these regions. Yellow perch especially seem to key in on these. Right at first ice, the ice shanties and ice fishermen will basically ring the areas around the Cisco spawning shoals because that's where the perch are. Alex Gatch and colleagues looked at Cisco diets. Cisco are consuming these eggs. We also actually see a lot of long-tailed ducks feeding in these regions as well in the fall after spawning. So as we think about sort of what's happening with these different processes and how they relate to habitat, we think that elevated sedimentation loads, right? So we cut all the trees down in these watersheds, 1840 or so. You get a big flush, but then you fill in all those river and stream valleys. And then you just get a constant press type manipulation of high sediment loads. We increase nutrient loads later in the sort of mid 1900s, and now you get a lot higher deposition from algal elements. And so we've really changed what the lake bottom looks like and the processes that occur there, so that if a fish depends on that, you know, we've really altered their system. With a wave action being obviously higher up shallow than it is deeper, you know, the fall is known to be a bit of an angry time in the Great Lakes. That also might pose an impediment in that you can actually have too much physical disturbance for those eggs that are up shallow. Then you end up in this Goldilocks zone where there's not a lot of physical disturbance, but there's still interstices to protect the eggs. Then you get down the bank where eggs can accumulate in big piles. Predators can easily get at them. Sediment loads are higher and substrates are sort of not ideal for egg incubation. You have ice come on, and you know, a lot of folks have seen the changes in year class strength with ice. We think that mechanism might basically effectively increase the area of the lake where substrates are suitable for successful incubation. In fact, Art Bonsel's master's work in 2017, where he looked at year class strength, found a really good correlation in Lake Ontario with earlier ice and larger year classes, very similar to Taylor Brown's work on the spatial patterns. And many folks have seen those patterns throughout the Great Lakes and the range of coregonines. So to really get at how we're going to disentangle that, we've set up a manipulative experiment where we want to change the substrate interstitial quality over a variety of depths to help disentangle the effects of physical drivers. So here in Chaumont Bay, we have 4 sites laid out where we have great before data, and we're going to try both cleaning those substrates and adding substrates to basically manipulate the interstitial quality at various depths, and then track deposition, settlement distribution, and emergence over those. And each one of those sites has control and treatment areas within the site. So this is sort of a Baki design that we're hoping this will really help us disentangle those different potentially competing or you know, mechanisms that are serving to work together to change the environment and how well a certain area can incubate coregonine eggs. In doing so, we think that'll help us make recommendations for if it's even feasible or possible to change substrate or remediate substrates to restore. So one of the neat aspects of this project is it's garnered a bunch of attention and excitement to do this in different places. No place is that probably more important than at the other end of the spectrum in Lake Superior, where, as many of you know, the sort of most intact coregonine populations are. There's still a big Cisco population up there that supports a roe fishery. The Apostle Islands region of Lake Superior there, where that red star is, all those islands, that's one of a known, very you know, consistent and big spawning area. And so we entered into this big collaborative effort with the Red Cliff Band and Wisconsin DNR and the Fish and Wildlife Service and National Park Service in that region to try to bring these methods to Lake Superior. And I say try because of any of you that have worked out there on Gitchigumi, you know she doesn't always take well to late season or early season sampling. But because of the great abilities of the crew, especially of the Wisconsin DNR and Red Cliff groups, we were able to set egg traps out in 2 different portions east of Madeline Island. A spot right off Madeline Island that dropped down off the shoal, off the point into deep water, about 60 meters there. That's the top red box. And then also a yet unnamed shoal that sort of peaks out at about 20 meters and drops down into 60 meters. A bit of an odd sampling design because there were a lot of gillnets when we were out there working. So we knew we were probably in and about the right place. So these next figures will just show maps, sort of the XY of where we caught these eggs. And again, we set these traps in October, and the teams up there pulled them in December. We set 186 and got 177 back. So we felt like that was a really good batting average for us. But again, we used the cutoff of 2.8 to differentiate Cisco sized eggs, right? Because they could have been a bunch of different coregonines from lake whitefish sized eggs. And when we do that, we see here the highest densities of Cisco egg deposition were off deeper. They were out in that 60-meter depth. And as you move shallower up either towards the shoal or the point, egg densities decline. And in fact, a lot of the 0 catches illustrated by the red X's there were up in those shallow habitats. Similarly, lake whitefish showed a pattern where their highest catches were up on those shallow areas, and then there were no eggs deposited or caught out deeper. And so that matches with what I think a lot of folks' conceptual model is for how lake whitefish and Cisco are spawning in these regions. If we bring in sort of now depth is on the x-axis and the type of substrate is on the y, we see this almost strong preference for a silt substrate of Cisco. I remember when Dan Yule taught me about the mud herring of Lake Superior and how that's what folks thought they were spawning over. And indeed, this evidence supports that model, although there is deposition in the shallower rocky areas as well, at least for Cisco. And that's where most of the deposition is for whitefish. So this stuff is just sort of really cool and, in my mind, interesting to think about. Like, what drives one population of Cisco to avoid that deep silt and the other to seemingly select for it? This is an old paper by Anderson and Smith Jr. from the 70s, where they were looking at lake herring in the western arm of Lake Superior. And there they were talking about how the fishery declined first in Duluth before it started to decline out by the Apostles. And there they mentioned that the sediment soil in the Duluth area contained a higher percentage of silt and organic matter than that in the Apostle Islands, very likely being driven by the landscape processes of the river that came into the harbor there. So they postulated that that might have something to do with the egg viability in the former area. So then we start thinking about sort of what we've done and how a lot of the embayments around the Great Lakes, you know, these really productive potential nursery areas, those are also the areas of the highest sort of maybe landscape impact and sedimentation. It might suggest that there's some sort of role there in this driver, you know, in sedimentation and habitat changes actually influencing the what we see is happening in the coregonine populations, both at a regional level, at a lake level, or at the entire basin scale. So with that, I will stop talking and see if there are any questions out there. And again, just want to take a nod and thank all the contributors, collaborators, and co-authors to this work. And thank you for your time.
Excellent. Thanks very much, Brian. Great talk. We will now You're seeing the virtual applause there. And for those that would like to ask questions, you can just throw your camera on. You can use the raise hand feature too, or you could even type your question in the chat. As people are thinking about that, one of the many observations you made was about our lack of knowledge, of course, about spawning behavior, given the time and place. And I know you've studied this around the world, like in terms of the literature. Do you have any sense of to what extent there might be staging by males, females coming in later, how deep or how shallow they might spawn to get at sort of are those eggs ending up where they intend for them to end up, or are they just sort of settling out along with all of the other, muscle hash, as you call it?
Right. Yeah, so Cam Davis did a really exhaustive literature search and got into Google Translate to translate Russian and a bunch of different languages and get at this. And we do have a nice analysis on that we're working up. The trends really seem to match what we've seen, both in the fact that there's not a lot of evidence lake-wide, although folks like Juha Karlainen and folks like that have really started to follow a very similar pattern in different lakes in Norway, Sweden, Finland. And so people are seeing that. They're seeing very similar patterns to what we're seeing where they are looking. But again, a lot of it is based on anecdotes. As for the staging, I think that's where a lot of the information that comes, especially from the commercial fisheries, is so valuable, because like up in Lake Superior, there's a constant information source about sort of the readiness of the Cisco there. They're trying to fish them for the eggs. And so they actually know they're not trying to fish them until they can get that right spawning condition. As for sex ratios, I'm not so sure. Your own work on sort of the skewness and sex ratios of bloater. And I thought we had a great idea of that in Lake Ontario, because we thought we see males coming in earlier and sort of exhibiting more aggressive behaviors. Not so sure that works out. It might be anecdotal. We might have just seen that because we saw that. But folks like Daryl Hondorp and Dan Yule's acoustic telemetry project, Dmitry Gorsky and Alex Gatch are working on a similar project. They'll actually have some great information to address that much. And I think there's probably other telemetry projects going on that if they have that sex information, that'll be key to understand those changes in behavior.
Great, thanks. Taylor.
I will happily ask a question. So before you showed the whitefish data for the Lake Superior sites, I was thinking about the role of physical disturbance and variability between Superior and Ontario that could have been maybe driving Cisco to spawn deeper in Lake Superior to avoid the most shallow, high physical disturbance regimes that might extend deeper in Superior. But then I saw the lake whitefish spawn shallower. And so I'm wondering what your thoughts are about the different physical disturbances between the two sites and what that might have implications for between the two species. Sure.
More disturbed is the easy way of putting it for Lake Superior, right? Like, I listened carefully and heard the stories and everything, but it really took being out there, in our 26-foot boat with leftover swells coming in from the north and a competing strong wind coming in from the east, to just be like, Aaron Heisey and Alex Beazle and I all kind of looked at each other. We're like, why did we decide to do this? Right. And it's like, this is exactly what Mark Vincent, Dan Yule, Sider, and all those guys have experienced for so long in trying to work out there in those conditions. It's just too damn hard and dangerous. Where we specifically picked it because we thought it was the sort of most protected area that we could get to close to a big spawning aggregation and not in a fishery. So we specifically picked that area because we thought it had lower disturbance, but is way, 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 way higher than anything we picked in Lake Ontario. So I don't know. I don't understand why, what is it about a whitefish that says, okay, I'll put my eggs up shallow, and in some places a Cisco says, yeah, I will too, versus deep, and how that segregation happens. And I don't know. There's a lot of neat sort of evolutionary pieces there. I don't have very many good answers for them now, but that's part of the reasoning for doing this, to throw this stuff out there, see what others see, think, call that. So it's definitely neat though. There's a lot of whitefish out there too. So
And where would, if you had lake trout, I mean, did you see any lake trout eggs in Lake Superior? Would those be intermediate or?
I'm gonna ask Aaron Heisey to check in on that. Aaron, do you know, did we see any eggs that we suspected were lake trout?
I don't recall seeing any eggs we suspected as lake trout. I know in some of the Ontario data there were a handful, but not in Lake Superior.
Yeah, okay. Hey, Ben.
Yeah, just have kind of a short question. If I can phrase this right. Do you think there's maybe some sort of a critical mass that goes along with these Cisco populations where they have to reach that, where some of these more less suitable spawning habitats become usable and they can deposit sort of this critical mass of eggs to kind of get over the hump with predators and that kind of thing, where some of these maybe more productive habitats like in Lake Ontario, where there's a little bit more protective, kind of become more important as these populations decline and get smaller? Maybe you can just kind of expand on that a little bit.
Sure, it certainly could. Right. I think if you're thinking too, the densities of egg deposition we saw in Lake Superior were 2 to 3 orders of magnitude lower than what we see in Lake Ontario, which is kind of counterintuitive given that we think the densities of coregonines are so much higher there. So are they that also just could have been because they're spreading eggs out over a wider area, right? So they're reducing the potential to sort of, they're making the predators work harder as well. So yeah, I think there could be that. I'd like to play out the physics though, and the abiotic and biotic attributes of this sediment. I know it, in just seeing it, it looks different what we saw out in 60 meters in Superior than what we saw in 60 or even 30 meters in Ontario. So there really might be a sort of an oxygen demand, a respiration element that differs among that then should really actually sort of feed on and control, why you would selectively not spawn out deep in a area that has a sort of elevated nutrient load. But I like what you're thinking. We should talk more about that at some point.
Let's see, let's go to Bruce, and then I'm seeing a few comments show up in the chat. We'll ask you those next. Bruce, please.
Yeah, I thought I saw or heard references to substrate additions for making improvements. What were people's ideas on that? What do you mean by that?
It's a good question. And I'll start by saying we're not sure. We're trying things. We're trying to learn from the work that's been done, like Michigan and like Huron. We're putting out, I want to say, we'll say fist size rock to try to specifically manipulate the interstitial quality in areas of already sort of hard regions, but that have been infilled, right, have already been embedded. So when we're adding it to change the condition and sort of experimentally test what effect that has. Certainly in other places, people have built big spawning reefs or lots of little spawning reefs. And so we're paying attention to those pieces. But we wonder were those places, was it as different? Was the substrate potentially as lacking as it appears to be in Lake Ontario, right? We can't really find interstices very well. And we've been looking all over. We've had folks looking for lake trout as well. And really it seems since Dracenids, there were some interstitial space when John Fitzsimons and Ellen Marsden worked on this stuff, a couple decades ago, but we may have lost those since we've got Dracenids. They could fill in the bigger cracks and their shells fill in every crack. So we're talking about adding substrate over sort of known rocky areas to just increase the interstitial element to see, because we think a lot of it could just be that the eggs are getting moved off of them. There's nothing to hold them. So if we can hold them, is that just the step that we need? And that plays into Titus's question of, do we just need to wait for another glacial advance? I don't know. Doesn't seem like we're getting one of those anytime soon, as warm as it seems to get around here. Historically abundant interstitial spaces. Definitely not going to get back to historical. The question is, can we manipulate that substrate at a scale that matters? I've got some bets out there already, and people are betting against us, and that's okay. I like that, because when you get to that point where people start to disagree, that's where we should be working. I have at my disposal a boat with 1,600 horsepower that we want to put prop wash deflectors on. I think we'll be able to move a substantial amount of sediment with that. And I think if we can just increase it a little bit, given how intense and how high the densities are in certain places, I think we might be able to have an effect. But that's what we're going about trying to test right now. Other folks think we can just stock rocks and that should work. We're going to try it all, but we're going to try it all and keep working on it. If we don't get it, we're going to try to figure out why. So
I know that I live in the Apostle Islands area. And so if you're looking for, let's say, a rock that is native to the areas, and it's like there's a construction project going on, and say, oh, by the way, I would love to have you dump it out here. Can't use artificial concretes and things like that. But if you move anything that's bigger, you attract a lot of lake trout, but that's not what we're after, Right. And so there you go.
Right. And all this, this stuff builds on the work that like Tom Binder and Chris Holbrook and them have done up in Lake Huron, and looking at how substrate, a big boulder and Farha, like what does a boulder do and how that maybe changes flow. And that's what the fish are targeting. We're trying to look at all those pieces and learn from all that as we attack this for a new species. But absolutely, we're really just scratching the surface and just getting into it, whereas a lot of the other lakes have explored this much more. That being said, we might have a more degraded system, so we might be able to have the effect size that with the manipulation that's not as evident in the lake that still has more interstices.
The more we learn, I guess, right?
Absolutely.
Thanks, Bruce.
All right.
Alex Gatch there.
Hey, Brian.
Yo.
That was awesome. I'm just trying to think about some stuff and bringing up out here. And my brain started turning about, some of the spawning strategies. And I think most people's conceptual models that lake whitefish are more bottom oriented. And when they are placing their eggs, so in Superior, where you were seeing those fish up in the rocky substrates and they're dropping eggs in that area, was different from the Cisco, that maybe the conceptual model is a bit different, more of a pelagic spawner, but still selecting for certain things. And I'm just trying to put together the differences that you see in Chaumont with Superior. Do you think, I know you've been trying to do this, I think the spawning strategy could be different in Superior. So maybe there's more of a pelagic spawn in those deeper areas. Substrate's not really an issue. So, or substrate isn't really their their target. It's more where the mass of the fish are spawning, more of like a pelagic spawning, where in Chaumont Bay, you're seeing more targeted towards rocky substrates or descended substrates. And so, you know, I mean, there's this plasticity within coregonines in general, but I was thinking about lake trout as well. Lake trout spawn on different types of substrates in different areas. Maybe the behavior of how they spawn is not different, but could you be seeing that among those between the populations in Superior and Chaumont as well, just a difference in how they actually spawn?
Absolutely. And that's another piece where, I mean, we've tried to start going about this with cameras and sitting out there in early December in the snowstorms and whatnot. It's not great conditions, but there's been precious little observations that can tell us even how, you know, contrast that with lake trout, right? Where, you know, we know the ins and the outs of it and, you know, finding new and unique behaviors. We still don't know. There's still debate among folks whether Cisco deposit their eggs low in the water column, middle, or high. And that was an important question. That was like, we can go after that. And we weren't prepared to answer that, but we see that as relevant for how these things settle. I will point folks, the literature, Vendace have been observed in a laboratory setting, and then a recent paper by Semenchenko and Smeshliva, I knew I destroyed those names, but talks about the spawning behavior of whitefish in Russia. And that's some of, in my mind, the best, most recent evidence. All of those whitefishes still, some changes, but still did just like the Vendace, did a pelagic release. So even if they were over 2 meters, they came up to the surface and released. If they were over deeper water, they still went up to the surface and released, but it could be slightly deeper. So absolutely, but without either the information from, say, acoustic receivers or video or Didson or something, it's hard to say exactly how that behavior could differ.
Yeah, more pie in the sky, but thinking about those hybrids of Lake Whitefish and Cisco and Eastern Lake Ontario, and that might explain more of a substrate driven or benthic driven spawning release in Chaumont versus
Sure
a more pure Cisco in Superior that might be doing something a bit different.
Right. Although there weren't, if I'm remembering Alan George's work, there's not a very high proportion of mules out there. It's really rare. I don't know how that compares to other systems, though. I don't know what
Sure.
rare is in context to other places. So
Certainly some of like what Alex was getting at, sort of the differences between Lake Superior and Lake Ontario results, you know, finding them out in the silt deep in Lake Superior, and then what Ben was saying with respect to maybe they've got to go out to that less optimal habitat, or maybe that's just more reflective of the diversity of a larger stock, that it's more intact in Lake Superior. I wonder if, I mean, do you know of any plans to go in and sample some of the more sort of formal like embayments within Lake Superior that might be more comparable like to a Chaumont Bay? I don't know if there is one, but to see if you're seeing similar patterns embayment to embayment.
Right. I think we're looking for volunteers. Does anybody want to put their hand up?
There you go. Our Canadian colleagues there in Black Bay, Thunder Bay. Yeah.
Well, they were the ones that got Matt Pavet out there, and they did some serious work to get that done in just tough conditions. Right. But, you know, when I talk to our administrators about this stuff too, that's important. You need a steel hulled boat, right? Like you need to do this safely. We're not going to do this in unsafe ways, or you need robots, right? You need Esselman's robot army to help us do this stuff or whatever. We have tried. Pete Esselman did try to fly the robot in such a way that we could try to near video and interpret where eggs were. didn't quite have the lights right for that, but we've been trying that because we know we have these places in Chaumont where there's just a lot of eggs consistently. So it makes a great place where we can test those different methods. But yeah, if anybody, I talked to some colleagues up on the North Shore in Minnesota that had tried to do some of this work and came back with everything destroyed. It can be pretty deflating too, right? I mean, we had an entire year of our gear in Chaumont, I want to say in 2018, the ice popped quicker than we could get to it, and it took all our gear. We're still finding remnants of our limno trash about the bay. So.
Okay. I couldn't find my hand. This is Lars. But what about going to smaller lakes? No, this has to do with how much wave action you have in these different systems. I remember Mendota, where the Cisco was spawning right up onto the shore of the what's it called, the beer drinking place there. What is it, Brian?
Isn't that everywhere?
Union. So I wonder if in a small lake where they really can spawn much shallower, if they will, and this is just no, these populations change over time.
No, absolutely. And the lake size piece there seems incredibly relevant to bring in and fetch, et cetera. If you, referring back to Hannah Schaefer's work, where fetch seems to have this defining role and where we've seen historic populations or not seen them. I will mention that I got involved with Bo and some of the folks in the Minnesota lakes, Coronas and Carlos, and trying to do some of this same trapping in those lakes where there's questions about UV exposures and differences and egg work. I referred to Nikki Berry's work before. We didn't do real well. I came in there thinking, yeah, I can help these guys. It was like, I need to help Pete Jacobson figure out where his Cisco eggs are. This is hot potatoes. Yeah, it didn't work. We caught no eggs. So yes, I agree. And if you want to go out to Mendota, we can definitely do that. But it takes someone with on the ground ability and knowledge to figure this out, because even the small lake logistics were pretty tricky to deal with the ice from a safety perspective. So
thanks.
Owen?
Yeah, I just have sorry, I was a little late getting in on tune here with the talk, but I have somewhat familiar with your work, Brian. I just had a question. Are you measuring oxygen levels at the surface in between the interstitial spaces, a few millimeters down into the sediments and so on in these habitats?
I wish we could, and that is something that we've wanted to do. We've tried to do, and I would argue we've been unsuccessful at doing it.
Right.
If you're familiar with that, the Ventling and Schwank papers, they talk about just how hard it is to actually get an accurate oxygen level in that very small boundary layer where diffusion actually starts to slow down. But that's the one where the egg experiences. So we've been to date unsuccessful at measuring anything other than basically 100% oxygen saturation, even when we did it in areas where we were pretty sure there would be an oxygen issue. So if folks have any way of knowing how to do that, we're all ears, because we think that could be a very obvious driver. We just haven't figured out. Do you have any ideas?
I mean, one of the things I just recall people making presentations about, say, Cisco eggs in Superior, and they as they go down in deeper water, their relative density changes to the point where they're essentially reach almost neutral density, so they don't really sink into the sediment.
Mhm.
And this has me thinking, you know, well, what are the kinds of things that Cisco could modify easily within their gene toolbox to change something like that? And this might be self-selecting, that if they did this over shallower water, their eggs might sink into the mud and die. But if they went into deeper water, they go down deep enough, they may not actually sink. And as you know, in Lake Superior, the water is really saturated down at the bottom. So I think part of it goes back to, well, their eggs are where they are because whatever the conditions are and whatever's been selected for, it works, because they keep coming back and doing it again and again. And the puzzle for us is trying to figure out how in the hell are they doing this?
Right. And how to measure the elements, that the key elements. So that, and that density piece about the eggs is really insightful, right? Like how things work at depth at 60 meters is far different than at 6, right?
Yeah, I have a hyperbaric chamber. You could actually at least go down 8 atmospheres with this and see what happens to egg density. And you could do it in a laboratory, actually, if you had the right equipment. And you had the eggs, of course. There's the other issue. But a hatchery would have eggs for you to play with.
Right, exactly. Well, and that's some of the stuff we're talking about based on, you know, what you've already been developing and things at Tunis. And I realized I was ignoring the chat. Good, Titus filled us in. It's the terrace. Bronte reminded us the size matters, and Trevor asked, hypoxia gene expression in eggs is a measurable snapshot. Interesting, as a way we could go get the eggs and look for that gene expression to see if they've experienced it. Interesting. I like it. Yeah, the potential for this is just sort of limitless, right? We were also thinking about sort of, now we have both. We have genetic evidence of who the parents are, what eggs they put down, and which larvae make it. You know, start thinking about what are the attributes of the fish that actually make, you know, larvae emerge.
The other thing that could be interesting in that sort of density question is, is there some variance in the lipid content of these eggs? Immediately you might think with older females that put out larger eggs and they're better fed, they may have a higher survival of their eggs in these environments because they don't sink into the mud. So there's a lot of interesting possibilities here. It also means that it could vary from year to year, which might be one of the factors in variable recruitment, too much high egg mortality. So it's I guess part of what I'm saying is that's an amazing place where fish and the environment can modify what's going on, and it affects the outcome of like where they spawn, where they most likely have success over time, etc.
Right. That's interesting. Wendy put hybrids in Chaumont, 3% from Chaumont Bay were hybrids. All but one were found in nearshore habitats. Interesting. I didn't think the number was that high from Ellen's work, but that might be from Wendy's stuff that predates that. So interesting. So, you know, it certainly made sense to us why you would see hybrids in Lake Ontario, given how similar the behaviors seem to be.
All right. Well, Brian, you generated a lot of good discussion there. Thanks. And that was thanks to your excellent presentation, as we expected. And again, as Brian thanked everybody, thanks for coming out on a beautiful Friday afternoon before your weekend starts. We had a really good turnout. I think as many as 66 people. Maybe we got higher, but that's a great turnout. And as I mentioned before, we'll try to pull this off again in July. We already do have one person that has been volunteered, and we'll see if we can get others. If you're interested, please let me know, because we'd like to give everybody the opportunity to have some exposure to share their results. Doesn't have to be polished, doesn't have to be finalized, can be sort of interim. It benefits all of us to to hear your ideas and sort of fertilize other ideas from outside of your lake or outside of even your continent, for those of you that have been joining us from Europe, for example. So with that, thanks to Nick for pulling this together, and thanks again to Brian, and we will see you next month.